Okay, 2 more minutes. We'll start at exactly 8 a.m. So we're just waiting for a few more peoples today. People, we are 29 pa rin. Kasama ako doon. Dalawa yung sa akin doon. Excited na para sa Wednesday, midweek holiday. Sige lang po, sir. Okay. So we can actually start in a few seconds. Okay, good morning again, everyone. So for today, we're actually going to be talking about fatty acid metabolism. Now, things to remember, or the things that we'll be covering for today's discussion would be fatty acid synthesis, fatty acid degradation, or what we call beta oxidation, ketogenesis as well. Okay. Now, okay, so before we start, let's just have a review regarding digestion and absorption of lipids. Now, we have already discussed this last time. So, remember that in terms of digestion in the mouth, it's more on chewing and breaking it down and mixing it with the saliva, okay, forming the bolus. And then with the enzyme lingual lipase, which is coming in from the cells from the tongue, there is already digestion or breaking down of the triglycerides into fatty acids and to the triglyceride or the glyceride backbone. Okay. Now, as it goes down into the stomach, there is already mixing and churning. So, yung mga muscles ng stomach, it's going to be mixing it up, breaking it down further. And then we have what we call the gastric lipase, okay, which is going to be available in the stomach, and this is also working on to the digestion of triglycerides. Now, there are cases that lipase are usually still active as the food reaches the stomach. Okay, but although they are working on it, but it's not going to be the full extent of the digestion process. Hindi siya ganun kalakas in terms of digestion. Then, after the small intestine, of course, after the stomach, it goes to the small intestine. Now, here, majority of lipids are undigested and usually are forming droplets. Now, we have one chemical or one enzymatic material that actually allows us to have this fats broken down even further, and you call it the bile. So take note that the bile is made in the liver. It is just stored in the in the gallbladder. Then it's released into the duodenum when there is need for it. Okay, in the small intestine. So take note of the locations on those particular parts. Okay, now it can be both hydrophobic and hydrophilic. So that's why they can interact both with fats and water. And it is actually made up of micelles of lecithin. So this is a fatty acid, phospholipids, cholesterol, bile salts, inorganic ions, and pigments. Kaya nga, di ba, if you, sino ba dito nakita na ng abdo ng bangus? May nakita na kayo ng abdo ng bangus? Yun yung tinatanggal nila, yun ang iniingatan ng mga nagtitinda ng isda na wag na mababasag dahil Ah, pag once na nabasag yan, kakalat na yan, di ba? Mapait, totoo yun. Mapait po siya. Okay? Tapos, so for those who are eating, sorry baka hindi yung iba sa inyo, hindi kumakain nung papaitan. Yan. Yung papaitan, ginagamitan siya ng abdo naman ng baka. So, kasama yung tuwalya, yung tinatawag nilang libro-libro, or actually those are the different stomachs of the cow. Tapos, hinahalo nila, nilalagyan lang nila ng ah, luya, yan, Diyos ko. Paboritong-paborito yan ng mga manginginom yan. Okay? Papaitan. Maasim kasi siya or mapait siya tapos sasabayan mo ng beer. Lakas tama yan. Tinuruan eh. Okay. Then it also is an emulsifier. So it means that they're actually breaking down large globules into small droplets. Remember when we had samples last time, 'di ba? Para ano, imagine niyo na lang, you have pieces of fat all together, and when bile is added them, they are broken down into slushies. O yung mala slurpy yung consistency niya. Okay? Slurpy fat. Then, um, once it's already emulsified, since it's already broken down to smaller pieces, it's now available for other enzymes to react. Kasi yung surface area nagbubasag na siya or lumiliit na yung surface area. And more surface area available, the faster the reaction it's gonna be, di ba? So another one is a pancreatic lipases. So this is also another lipase that is found from the pancreas, and this is usually sent to the small intestine to help digest the triglycerides. Now, your triglycerides are broken down to fatty acids and monoglyceride. So, pag sinabi mo monoglyceride, you have one fatty acid still. Now, why is it's still it's not completely broken down? Kasi it can still enter. But as a triglyceride, it cannot enter the actual system as a whole. Okay? It's always a part of it. Okay? Or partially digested. Okay. Now, in the small intestine, this is where we usually absorb your lipids. So usually you have your fatty acids, your monoglycerides, cholesterol, and other fat-soluble vitamins are now available for absorption. So with bile, so, di ba kanina naka-cluster sila, they are now broken down and usually grouped that together with what we call the micelle, and this is actually able to get the fats closer enough to the microvilli so that they can be absorbed. Okay, and once the products of the fat digestion moves across the membrane on the intestinal cell, balik ulit sila to form the micelle. So micelle broken down, then micelle ulit sila. Okay. So inside this small intestine, your short and medium fatty acid chains and glycerols can be absorbed into the bloodstreams. However, yung mga very large natin na lipids, like yung mga long fatty acids, monoglycerides, and such are usually uh, need assistance. So they usually form another compound or another micro molecule so that they'll be able to pass through the bloodstream. Okay? And then, yung ating, yung sinasabi ko sa inyo, our fatty acids and monoglycerides, once they have passed through the intestinal cells, they are reassembled. Okay? And then, they now form what we call the chylomicrons. Ito yung mismo, this is actually the transport vehicle for your long chain fatty acids and the other materials that were not easily absorbed by the bloodstreams. Okay? Yan. So here is just a graphical representation of our enzyme action, okay, or our fatty acid absorption. Okay, so ito yan. So, your fats are broken down by bile. Okay. And then, afterwards, it's now going to be broken down into monoglycerides and fatty acids. Okay. So, once it's already formed now, your globules. once it already passes the intestinal villi, this is actually where some of them are being absorbed, some are going to form your chylomicrons that will be entering our lymphatic system. Okay, yeah. So just to give you an overview, so I have here a video showing the model of the plasma, triglycerol, and cholesterol transport in the humans. Okay. So again, this is one of those videos that I was able to download back then and was able to save it, but it's not easily available on YouTube. So kindly watch na lang po. Wait lang. Okay, so wait lang po. So again, I forgot this does not have any audio. So yan, explain lang natin yan. So here is actually a video showing how your fatty acids are moving inside the body. Okay, so yan. So chylomicrons bind dietary triglycerols and cholesterol in the intestine. So in the bloodstream, chylomicrons bind into the endothelium of the capillaries of skeletal muscle and adipose tissue. And the enzyme lipoprotein, oh, sorry, ang bilis. And the enzyme lipoprotein lipase hydrolyzes the triglycerols and free fatty acids. Okay, so you can see here, chylomicron remnants, mostly cholesterol return from the capillaries. Okay, so nabawasan na siya ng fatty acids. Yan. So the liver takes up the chylomicron remnants to a receptor and dietary cholesterol is delivered. Bile acids and cholesterol are delivered from the liver to the intestine. Then the liver is also part of the endogenous pathway of lipid transport. Remember we discussed VLDL. So they are synthesized in the liver and they deliver endogenous triglycerols and cholesterol to the tissue. So the VLDLs are dilapidated the capillaries by action of lipoprotein lipase. Ayan, to release free fatty acid, which are taken by the cells of the muscle and adipose tissue. The glycerol backbone is delivered to the liver or kidney cells to be converted to dihydroxyacetone phosphate. Dilapidated VLDL emerge from the capillaries as intermediates of IDL, intermediate density lipoproteins. So this is already your IDL. Okay. So after the degradation of to IDL or LDL, half are taken by the liver via receptor-mediated endocytosis, and tissues on the other than the liver take up cholesterol from LDL via LDL receptors. Cholesterol is removed from the cell surface, remember, so by HDL. This cholesterol is delivered. Oops, sorry. Sorry, let me just go here. Yan. So this cholesterol delivered to the liver by a process that's still under study right now, sabi lang niya. Okay? So yan. Okay. Any questions so far, guys, before we proceed? Clarifications, violent reactions? Rika, Nojana, questions? Thank you, Miss Asuncion, for clarifying. Okay, so let's move on forward. Wait lang. Ayan. So let's talk about fatty acid synthesis. Okay, for fatty acid synthesis, this is actually again one of those screen grabs that I got from osmosis.org, which is a good source of learning materials as well. So I also added the YouTube link here, although it's not publicly available. Okay, so yeah. Yan, nagbabuffer pa siya. Wait lang po.
In addition to carbohydrates and proteins, lipids are the third main macromolecule we consume in our diet. Fatty foods include red meat, dairy products, and even peanut butter. And lipids come in many forms, including cholesterol, glycerol, phospholipids, and fatty acids. Of these, fatty acids are the simplest form of lipids. They're basically just long chains of carbon and hydrogen that are grouped by length into short, medium, long, and very long chain fatty acids. Fatty acids can also combine with glycerol to make triacylglycerides, or just triglycerides, which is made of three fatty acids attached to a glycerol molecule. And this is the main storage form of fat in our body. Now, short and medium chain fatty acids are primarily obtained from the diet, but the liver and fat cells can synthesize long chain fatty acids. This happens by combining lots of two carbon molecules called acetyl coenzyme A, or just acetyl CoA, into a single 16 carbon long chain fatty acid called palmitoyl coenzyme A, or just palmitoyl CoA. Palmitoyl CoA can then serve as a precursor to even longer chain fatty acids. To make palmitoyl CoA, acetyl CoA provides the carbon atoms, and nicotinamide adenine dinucleotide phosphate, or NADPH, provides the hydrogen atoms. As it turns out, most of the acetyl CoA used to make fatty acids comes from carbohydrate metabolism, specifically glucose, which is a 6-carbon sugar molecule. After eating a glucose-rich dinner, like cake and cookies, glucose levels in the blood rise quickly. In response, the pancreas secretes insulin, which is a hormone that makes our cells take in and process a lot more glucose. Inside the cells, glucose can enter glycolysis, where it's broken down into two 3-carbon pyruvate molecules. And that yields a bit of energy in the form of adenosine triphosphate, or ATP. Pyruvate then moves into the mitochondria and is converted to acetyl CoA by an enzyme called pyruvate dehydrogenase. Inside the mitochondria, acetyl CoA enters the citric acid cycle by combining with a molecule called oxaloacetate to form citrate. Citrate can then continue in the citric acid cycle, which generates electron carriers that can join the electron transport chain in oxidative phosphorylation. All of this leads to the formation of a lot more ATP. So the math is simple: more glucose, more ATP. Well, ATP actually inhibits some enzymes in the citric acid cycle, which slow it down overall. And that means that extra acetyl CoA can be used to make fatty acids instead. The enzymes required for fatty acid synthesis, though, are all in the cytoplasm. So in order to start fatty acid synthesis, acetyl CoA needs to get out of the mitochondria. Unfortunately, acetyl CoA cannot cross the mitochondrial membrane. So to get to the cytoplasm, it combines with oxaloacetate to form citrate, just as it would to enter the citric acid cycle. So when there's a lot of ATP around, citrate crosses the mitochondrial membrane and enters the cytoplasm. In the cytoplasm, an enzyme called citrate lyase cleaves citrate back into acetyl CoA and oxaloacetate. This process of conversion and reconversion is called the citrate shuttle. In the meantime, oxaloacetate is recycled and goes back into the mitochondria so it can be available for the next incoming acetyl CoA. But we have another problem. Oxaloacetate can't cross the membrane either. So an enzyme called malic enzyme converts oxaloacetate into pyruvate, forming NADPH from NAD+ in the process. Now pyruvate can cross the mitochondrial membrane, and an enzyme called pyruvate carboxylase converts it back into oxaloacetate, which can start a new cycle. High levels of acetyl CoA can increase the activity of pyruvate carboxylase, so that oxaloacetate is made available. With acetyl CoA in the cytoplasm, all we need to start fatty acid synthesis is NADPH to provide the hydrogens. Some of that NADPH is generated by malic enzyme when it converts oxaloacetate to pyruvate. And the rest comes from the metabolism of excess glucose in a pathway called the pentose phosphate pathway. Once there's enough NADPH, acetyl CoA can start its journey towards palmitoyl CoA. All right, so first off, a carboxyl group is added to acetyl CoA by an enzyme called acetyl CoA carboxylase, converting it to the 3 carbon malonyl CoA. This enzyme requires 3 cofactors, which can be easily remembered by the mnemonic ABC. A is for ATP, B is for biotin or vitamin B7, and C is for carbon dioxide or CO2, which is the source of the carboxyl group. This additional carbon, though, will not contribute to the fatty acid chain because it's lost later on. This is considered the rate-limiting step of fatty acid synthesis, which means that the speed of this reaction will determine the overall rate at which all of fatty acid synthesis happens. So acetyl-CoA carboxylase is tightly regulated. There are 2 types of regulation: hormonal regulation and allosteric regulation. Hormonal regulation involves the pancreatic hormones
Okay, wait lang guys, nagbabuffer siya.
insulin and glucagon, and they work by adding or removing a phosphate group on acetyl-CoA carboxylase. When insulin is released, like after that cake and cookie bonanza, it activates the enzyme protein phosphatase 2, which removes a phosphate group from acetyl-CoA carboxylase, increasing its activity. On the other hand, when glucagon is released, like when you're fasting, it activates an enzyme called adenosine monophosphate, or AMP, dependent kinase, which adds a phosphate group to acetyl-CoA carboxylase, decreasing its activity. So glucagon basically handcuffs the enzyme, and insulin sets it free. This makes sense, since you want to break down fatty acids for energy when you're fasting, not use energy to build them up from scratch. The other type of regulation is allosteric regulation, and that's when a molecule increases or decreases the activity of an enzyme by binding to a different site on the enzyme than the site where the substrate binds. Citrate allosterically increases the activity of acetyl-CoA carboxylase, while fatty acids allosterically inhibit it, signaling that we have enough fatty acids and we don't need to make more. Alright, here comes the nitty-gritty part. In order to polymerize our acetyl-CoA monomers, we need an enzyme complex called the fatty acid synthase complex. Overall, the enzyme complex is made up of a lot of different enzymes and looks a bit like a kidney bean. The complex has 2 separate binding domains at each end of the bean. On one end, there's an acyl carrier protein, or ACP, and on the other end, there's an enzyme that has a cysteine amino acid in a very exposed position. The ACP is where acetyl-CoAs and malonyl-CoAs bind initially, and the cysteine amino acid residue is where they hop on board the growing lipid. It all starts when an enzyme that's part of the fatty acid synthase complex called acetyl-CoA ACP transacylase removes a CoA group from the acetyl-CoA molecule. The enzyme then attaches the 2-carbon molecule acetate to ACP, and then acetate spontaneously hops onto the cysteine residue. Now that acetate is bound to the cysteine residue, the ACP residue is empty. So it's time to bring over that initial malonyl-CoA that we made in the first step of fatty acid synthesis. Once more, there's an enzyme that's part of the fatty acid synthase complex called malonyl-CoA ACP transacylase, which removes a CoA group from the malonyl-CoA molecule. The enzyme then attaches the 3-carbon molecule malonate to the ACP group. Now we have a 2-carbon acetate on the cysteine end, and a 3-carbon malonate on the ACP end. At this point, another enzyme in the fatty acid synthase complex called 3-ketoacyl ACP synthase does 2 important things. First, it cuts off the carbon that acetyl-CoA carboxylase previously added and releases it as CO2, leaving behind a 2-carbon acetate. Then 3-ketoacyl ACP synthase moves this acetate and condenses it with the acetate molecule attached to the cysteine group. 2 carbons on top of 2 carbons gives us a 4-carbon fatty acid chain attached to the cysteine group. These 2 reactions require hydrogen from 2 NADPH molecules. This cycle repeats again when the next incoming malonyl-CoA molecule attaches to the free ACP group. And it happens for 7 cycles, until we've got a 16-carbon long fatty acid polymer. Per cycle, we need 1 acetyl-CoA to be converted into malonyl-CoA, and 2 NADPH molecules. But in the first step, we placed an additional acetate molecule on the ACP group, instead of converting it to malonyl CoA. So, summing it up, to make the 16 carbon palmitoyl CoA, we need a total of 8 acetyl CoAs and 14 NADPH molecules. Once the fatty acids are made, they're stored in the liver and fat cells as triglycerides. And when they're needed, they can be broken down in order to make ATP. Alright, as a quick recap, fatty acids are energy rich carbon compounds that are synthesized in the cytoplasm of liver and fat cells, and are regarded to be one of the main forms of energy storage. Fatty acid synthesis is the conversion of 2 carbon acetyl CoA molecules into the 16 carbon palmitoyl CoA in the presence of NADPH. The three major steps of fatty acid synthesis are the citrate shuttle, acetyl CoA carboxylase, which is the rate limiting enzyme, and finally the fatty acid synthase complex.
Okay, so that is for fatty acid synthesis. Okay, so I'll add the link later on and I'll have it saved in our TBL Hub as a PDF file so that you can actually download or watch the video on your own there. Okay, so this time let's talk about fatty acid. By the way, before we proceed, any questions or clarification regarding fatty acid synthesis? Clarification, guys? Sure, John. Clarification, Sir John? Questions? None. Thank you, Rika. Thank you, Miss Milan. By the way, we have my department head, my beloved Dr. Aldea on our session today. Good morning, ma'am. Okay. So let's talk about fatty acid degradation. Okay. So for fatty acid degradation, this is actually uh, a was discovered by Franz Nook. Okay? So, what he did is actually, may konting background story tayo pagdating sa mga gatong bagay-bagay. Mahilig ako sa mga maliliit na chismis. Okay? Small, meaty information that would actually help in the study. Okay? So, Franz Nook was able to feed his dogs with fatty acid, which is in which is usual terminal methyl group had a phenyl group. So usually with the phenyl group, he was able to determine what would be the output. So he had actually labeled the gamma fatty acid, and he was able to isolate the metabolized fatty acid from the urine. Okay, and what he found is that for even numbers, the products were phenyl acetate and acetate. Okay, so take note, guys. Yan, so uh, for even numbered carbon atoms, it will give rise to product phenyl acetate and acetate. Now, when he changed it to benzoate, sorry, not benzoate, but odd numbered fatty acids, he was able to form benzoate as one of the products and acetate. Okay? So yan po. So take note of the products that would be produced when you're undergoing uh, degradation. So pag even, one of the output is phenyl, acetate, and acetate, while for odd number, it's gonna be benzoate. Okay? Kasi syempre, yung excess po will just, if you will not see the structure, meron kang kasing extra methyl group here. Ito wala na po siya dahil nga odd number siya. Kumbaga, sumakto lang siya dyan. Okay. So we call it as beta oxidation. So this is actually happening inside the mitochondria and it is aerobic in its process. Now, we actually break down the fatty acid into acetyl coenzyme A, and the acid moves to the pathway as coacyl CoA derivatives using NAD and FAD uh, intermediates. Now, in order for it to happen, the fatty acid needs to be activated utilizing ATP in the presence of methyl coenzyme A or CoASH and acyl CoA synthetase. Okay. So the long chain acyl-CoA enters mitochondria bound to carnitine. Kaya nga, remember kanina dun sa video, there's a movement, okay, from sa mitochondria to the cytoplasm. Dito naman tayo. For it to happen, since it's going to be inside the mitochondria, it has to be shifted back from sa cytoplasm towards the mitochondria. Okay? So usually it's going to be bounded to the carnitine. Kaya if you're going to look this up, you will see that they usually refer to this particular part as what we call the carnitine shuttle. Okay? Kasi nga they shuttle the fatty acids inside and outside the mitochondria using the carnitine. Okay? So inside, once it's already inside, beta oxidation already takes place in which two atoms of carbon are removed simultaneously to form acyl coenzyme A and acetyl coenzyme A at the carboxyl terminal. Now here, so during this event, the bond is broken between the second carbon, okay, which is the actually labeled as the beta carbon. That's why it's called beta oxidation, because the bond between the beta and the gamma is gonna be removed. Okay? So yan, kaya nga beta oxidation siya. So this is actually going to provide energy from the fats. Now, so let me just go over the steps. Then later on, we will actually show you the video on how the process is. Okay. So the first step is the activation reaction that results from the production of fatty acyl coenzyme A molecule. So usually you have a thiester bond that is formed between the coenzyme A and the fatty acid, and it requires ATP. Okay. And it's cleaved to produce AMP and pyrophosphate. Now, CoA is required for this step, and the product is fatty acyl CoA, and has a high energy thioester bond between the fatty acid and CoA. So this one is an acyl CoA ligase, which is the enzyme that catalyzes the reaction, and this is located in the outer membrane of the mitochondria. So here is the reaction. So you have the fatty acid, then you actually add the coenzyme A with ATP. Now, take note, hindi lang siya isang phosphate ang tinanggal, dalawa po siya, causing them now to form a thioester bond between coenzyme A and the fatty acyl. Okay, kaya ang pangalan niya ngayon is fatty acyl coenzyme. Fatty acyl CoA na siya. Okay? Now, in this part, the mechanism that brings the fatty acyl CoA into the mitochondria usually involves a carrier molecule carnitine. So this is the actual carnitine shuttle. So it's catalyzed by the enzyme carnitine transferase 1, and it transfers the fatty acyl group to carnitine. So as you can see here, it's attaching itself to the hydroxide here, producing acyl carnitine. Okay, so ito po siya ngayon. Now, so here is the actual movement now of your acyl carnitine. So the carrier protein is located in the mitochondrial inner membrane, transfers the acyl carnitine into the mitochondrial matrix, and then carnityl acyl transferase 2, okay, catalyze the degeneration of fatty acyl CoA, which now becomes involved in the remaining reactions of beta oxidation. So ito si translocase. Okay, so transfer siya doon. And then you have your carnitine acyl transferase. Okay, so here is your carnitine acyl transferase. So this is the one that is causing it to go inside the mitochondrial matrix. And then afterwards, hiwalay na si acyl coenzyme A and carnitine. Then carnitine is shifted back into the cytoplasm. Okay? Parang ano lang siya, delivery. Okay? Now, the next reaction is the oxidation reaction that removes the pair of hydrogen atoms from the fatty acid. Okay? This used to reduce FAD to FADH2, and then the dehydrogenated reaction is catalyzed by the enzyme acyl-CoA dehydrogenase, and results in the formation of carbon-carbon double bond. Okay, so here on this particular part. Then oxidative phosphorylation yields two ATP molecules for each FAD. Okay, take note here, this is happening inside the mitochondria. The conversion of FADH is to ATP, and we discussed this already in electron transport chain. Okay. Now the third step, so this one involves the hydration of the double bond produced in reaction 2. So as a result, the beta carbon is hydroxylated, and the action enzyme is enzyme enoyl coenzyme A hydrase. Okay, so we're actually going for this one. The water molecule will break down the double bond. Hydroxide is attached here at yung isang hydrogen dito. Okay, so sa organic we call this as Markovnikov's rule. Okay, pero we're not gonna go into that one. What happened here is that we actually add water into the system to break down the double bond. Okay, and part 4, the reaction of hydroxyl group of beta carbon is now dehydrogenated. So NAD1 is reduced to form NADH that is subsequently used to produce 3 ATP using oxidative phosphorylation. Okay, and you have there the enzyme beta hydroxyacyl CoA dehydrogenase. So here, what happened is that kanina, di ba, double bond siya dito. Once we made this as a hydroxide, pwede natin siya nang gawing double bond. And then the hydrogen is the one that's going to produce now your NADH by reducing the NAD ion. Okay. And the final reaction here using the enzyme thiolase, we cleave now the acetyl coenzyme A and producing now your acetyl coenzyme A. Okay, so here, so what will happen is that we're actually going to remove it na, okay, from our beta carbon. Okay. So this actually shortens the fatty coenzyme A, continuous process until you only have 2 to 5 components or carbon molecules. And then the rest are completely degraded to acetyl coenzyme A. And this is how we produce acetyl coenzyme A from fats. And this coenzyme A now is going to enter now the TCA cycle. Okay? Now, here, oh, I got here another reminders. So in case if it's even a numbered acyl coenzyme A, beta oxidation enters 4 carbon acyl coenzyme A broken down into 2. Okay, so each one containing 2 carbon atoms. So if it's uneven, definitely it's going to be full acetyl coenzyme A. However, if it is odd, one of them will become propionyl coenzyme A. And the rest is 2 carbon acetyl coenzyme A. Okay? So, tandaan ninyo. So, for beta oxidation of fatty acyl, it's if it's even, everything will be acetyl coenzyme A. However, if it is odd, one propionyl coenzyme A, then the rest is coenzyme A. Okay, so here is the overall reaction. So take note. So you have here your acyl coenzyme A. Take note of the number. Okay, so your FAD forms your FADH2, NAD forms NADH, then water is broken down into hydrogen ion, then coenzyme A is usually bonded to acetyl CoA and acyl CoA. Okay, so take note of the numbers here. So if you have, again, as mentioned earlier, if you have even, you're gonna be -2. Okay, yeah. And if it is odd, it's gonna have a difference. Yung isa sa kanila is gonna be propionyl coenzyme A. Okay, so yan. So here now is beta oxidation of palmitoyl coenzyme A. Okay, so teka, tanggalin ko. Wala siya talagang audio. So again, this is one of those technologies that were made when Flash was, or yeah, Flash Weaver was available. Ngayon wala nang Flash Weaver, tinanggal na siya. So screenshot grab ulit siya. Okay? So here is step by step ng explanation ko kanina. Okay? So you have already a fatty acyl coenzyme A. Okay. So the action of FADH. So if you will notice, yan. So this is the part wherein we produce a double bond between the alpha carbon and the beta carbon. And then the hydrogen from this part produces your FADH2 or reduces it to FADH2. Okay. Then the next one, you have now water molecule, removing the double bond. Yan. And then the next one, you have now NADH. Tama ba? Saan na si NADH? Yan. So NADH, reducing it to form NADH. NAD lang pala. NAD to NADH. And this one causes a double bond with this particular carbon, with a beta carbon, and releases one hydrogen ion. And this is the time when we're going to be adding now coenzyme A to the to the alpha carbon, producing now one acetyl coenzyme A. So that's the first cleavage. Okay? So we will continue on with this process until we have already consumed all of the material. So as you will see here, as it goes on, it becomes faster. Yeah, so FAD ion, double bond with the beta. Then you have water, removing the double bond, forming now your hydroxide here. NADH, NAD to NADH, forming double bond oxygen or carbonyl. And then coenzyme A bonding to the alpha, releasing acetyl coenzyme A. So as you can see here now, it's getting faster and faster. And that's the process of beta oxidation. Okay. Okay, so as you can see, it's continuously happening hanggang sa maubos siya. So, nakailang cycle na tayo. So, this is fourth, going to the fifth cycle. Okay, so this is actually, tama ba? Hindi ba last to? Ayan. Okay, so you have now the whole acetyl coenzyme A formed from our uh, palmitoyl coenzyme A. Okay, so if you go recall, if I go back here and pause it a little bit, if you will notice that is palmitoyl is 18, right? 18 carbons. And then you only produce 8 is acetyl coenzyme A. Okay. 2, 3, 4. Wait lang. Forward natin para yung kompleto ang makita natin. Hindi ko na naman nakuha. Yan. So you have there 8 acetyl coenzyme A, then you have 1, 2, 3, 4, 5, 6, 7. Okay. So 8 ko A, then 7 yung cycle. So it means 7 times tayo na nagputol to form our acetyl coenzyme A. questions, clarification, guys, before we proceed to the next one? Elora, clarification. Okay. Thank you, Ray. Thank you, Miss Katinoy. Okay, so let's go to the next one. Okay, so this one is again from osmosis.org.
Our bodies are capable of surviving without food for long periods of time, up to 2 weeks. The reason we can do this is that we can store our dietary fuels and then break them down when we need them, to make energy in the form of adenosine triphosphate, or ATP. Fat is one of the most important ways that we store energy, and the term fat burning actually refers to fatty acid oxidation. In fact, if 2 individuals were stranded in the Andes Mountains with no food, the person with more fat content would survive longer. Yet another reason to avoid working out. What makes fat such a great source of energy are the fatty acids, which are the simplest form of fats, composed of long chains of carbon and hydrogens. The transfer of electrons in the form of hydrogen molecules from these fatty acids to certain molecules can then be used to generate ATP. Fatty acid oxidation primarily takes place in the mitochondria of the heart, skeletal muscle, and liver cells. Before we can oxidize fat, it needs to be moved from storage sites to the cells that can use it. Fat is stored in adipocytes or fat cells as triglycerides, which are 3 fatty acids attached to a glycerol molecule. Triglycerides can be broken down by the enzyme hormone-sensitive lipase into 3 fatty acids and glycerol. So if you're starving in the Andes, first your blood glucose level falls, and in response the pancreas secretes a hormone called glucagon, which increases the activity of hormone-sensitive lipase, and increases the breakdown of triglycerides. Now, the free fatty acids can leave the fat cell and enter the bloodstream, where they bind to a protein called albumin. Albumin carries the fatty acids to target cells, like liver cells, that are capable of fatty acid oxidation. First, the free fatty acid dissociates from albumin and diffuses into the cell. Once inside the cell, a cytosolic enzyme called fatty acyl-CoA synthetase adds a coenzyme A molecule to the end of the fatty acid, turning it into a metabolically active fatty acyl-CoA. This process requires 2 ATP molecules, so it takes a little energy to make energy, but it's totally worth it in the end. Now, the mitochondria is composed of 2 membranes, an outer membrane and an inner membrane, with a small space in between, and the mitochondrial matrix at the core. The enzymes required for beta oxidation are located in the mitochondrial matrix. However, the fatty acid can't cross the inner mitochondrial membrane when CoA is attached to it. To get around this problem, we need to free the CoA from the fatty acid. An enzyme within the outer mitochondrial membrane called carnitine acyltransferase 1, or CAP1, replaces the CoA with a carnitine, making fatty acylcarnitine and a free CoA, both of which can easily cross the inner mitochondrial membrane. Then, along the inner mitochondrial membrane, another enzyme called carnitine acyltransferase 2, or CAP2, substitutes carnitine and CoA back, therefore regenerating fatty acyl-CoA and free carnitine, which is now within the mitochondrial matrix. This whole slick process is called the carnitine shuttle. Carnitine can cross the inner mitochondrial membrane by itself, so it can go back to the outer membrane to meet the next incoming fatty acid. We can also get more carnitine from our diet, mainly meat products. Finally, the carnitine shuttle can be regulated by a product of fatty acid synthesis called malonyl-CoA, which specifically inhibits CAP1, slowing down fatty acid oxidation. After all, you don't want to make and break fatty acids at the same time. Okay, so fatty acid chains can vary in length. We've got short, medium, long, and very long fatty acids, but the process of fatty acid oxidation is the same for any length. We'll explain fatty acid oxidation with an example of a long-chain fatty acid, the 16-carbon long palmitoyl-CoA. The enzymatic modifications that take place in fatty acid oxidation happen on the second and third carbon of the chain, also called the alpha and beta carbons respectively. Each of those carbons enters the fatty acid oxidation with 2 hydrogens bound to it. First, an enzyme called acyl-CoA dehydrogenase removes 1 hydrogen from the second carbon and 1 hydrogen from the third. The same enzyme then gives those 2 hydrogens to a nearby flavin-adenine dinucleotide molecule, or FAD, making FADH2, and converting the palmitoyl-CoA to enoyl-CoA in the process. Enoyl CoA has one hydrogen on each of the alpha and beta carbons. This is the first oxidation step. Next, an enzyme called enoyl CoA hydratase transfers a hydroxyl group, which is an oxygen linked with a hydrogen from a water molecule, onto the beta carbon of enoyl CoA, making beta hydroxyacyl CoA. The second oxidation step involves a similar hydrogen transaction mediated by another dehydrogenase called beta hydroxyacyl dehydrogenase. Beta hydroxyacyl dehydrogenase removes two hydrogens from the beta carbon and transfers one of them to a nicotinamide adenine dinucleotide molecule, or NAD+, making NADH and beta ketoacyl CoA in the process and releasing the other hydrogen. And because this oxidation step occurs at the beta carbon, fatty acid oxidation is also sometimes called beta oxidation. Finally, an enzyme called beta-ketothiolase cleaves those two alpha and beta carbons off the fatty acid chain, making a 2-carbon acetyl-CoA molecule in the process. This leaves us with a 14-carbon fatty acid chain that can enter another cycle of beta oxidation. So the longer the fatty acid chain, the more cycles and the more energy we make. And in one cycle of beta oxidation, we make one NADH, one FADH2, and one acetyl-CoA. NADH and FADH2 are now full of electrons, and they can enter the mitochondrial pathway called the electron transport chain, where those electrons can be used to make ATP. One NADH molecule makes roughly 3 ATPs, while one FADH2 molecule makes roughly 2 ATPs. On the other hand, acetyl-CoA can enter the citric acid cycle, which is another metabolic pathway in the mitochondria that burns acetyl-CoA to make more NADH and FADH2, which then enter the electron transport chain, yielding a total of 12 ATPs per acetyl-CoA. So from a 16-carbon palmitoyl-CoA molecule, we make a total of 7 NADH, 7 FADH2, and 8 acetyl-CoA molecules. If we calculate the total ATP yield, we make 131 ATPs. But remember, we consumed 2 ATPs when we first activated fatty acyl-CoA. So per palmitoyl-CoA, we make a net worth of 129 ATP molecules. That's a lot of ATP. I told you it was worth it. Now, that's straightforward enough for a fatty acid that has an even number of carbons. But the oxidation of a fatty acid with an odd number of carbons can be somewhat different. So let's say we're dealing with a 13-carbon fatty acid. Initially, the process of oxidation goes exactly the same, until we reach a 3-carbon fragment called propionyl-CoA. This is when it gets a bit awkward for the prototypical oxidation enzymes. So to get the job done, we'll need another 3 enzymes to break it down. 3 reactions for the 3-carbon molecule, if you like. So first, an enzyme called propionyl-CoA carboxylase adds a carboxyl group to the propionyl-CoA, making methyl-malonyl-CoA. This enzyme requires 3 cofactors, which can be remembered with the handy mnemonic ABC. A is for ATP, B for biotin or vitamin B7, and C for carbon dioxide, which is the carboxyl group source in this reaction. Next, an enzyme called methyl-malonyl-CoA mutase rearranges the carbons on methyl-malonyl-CoA to make succinyl-CoA. This enzyme requires vitamin B12 as a cofactor. Succinyl-CoA can then enter the citric acid cycle, or it can be used for the synthesis of heme, which is the oxygen-binding element in hemoglobin. Finally, for really long fatty acid molecules that are 22 carbons long or longer, specific organelles called peroxisomes may be needed. Peroxisomal oxidation works in the same way as mitochondrial oxidation, but it just has different enzymes that are capable of degrading the long fatty acid chain until it's under 22 carbons, and then the fatty acids are sent back to the mitochondria to take over. Alright, so, as a quick recap, fatty acids are one of the main sources of energy in the body, especially during the fasting state. Fatty acid oxidation occurs in 3 steps: activation, then transport through the carnitine shuttle, and finally beta oxidation in the mitochondrial matrix.
Okay, sorry guys. It's 16 carbols of palmitoyl or palmitic acid. So here, as mentioned in the video, what we're looking at right now is the overall of the palmitic acid. So the activation to palmitic acid to form palmitoyl acid, coenzyme A, and then it enters your beta oxidation, releasing 8 acetyl coenzyme A that enters citric acid cycle. Okay, so overall in all, you get here 129 ATP. Okay, so you have there coming from the ATP from FADH and ADH as well. Okay, any question, guys, or clarification? Mr. Ortiz, any questions? Rika? None. Okay, thank you, Rika. Okay. So let's talk about ketone bodies. So usually for ketone bodies, you have your acetyl coenzyme A, which was actually produced earlier as discussed in beta oxidation to efficiently enter the TCA cycle. And there must be an adequate supply of as oxaloacetate. Now, however, if glycolysis and wet oxidation are occurring at the same time, there would be a steady supply of pyruvate from glycolysis that can be converted to oxaloacetate. Now, if the supply of oxaloacetate is too low to allow of all the acetyl-CoA to enter the TCA cycle, your acetyl-CoA is now converted to ketone bodies. So your ketone bodies are usually acetone, beta-hydroxybutyrate, and acetoacetate. Okay. Now, ketosis is this is actually the metabolic state characterized ano ba yan, by elevated levels of ketone bodies. I think you might have heard this this is actually a trending diet right now. Di ba? Sabi ko sa inyo, I've told been over and over that biochemistry is the best friend of dietitians, cosmetic surgeons, at saka yung mga nag-a-apply ng mga beauty products sa katawan nila. Okay, so keto diet. Is there anyone here who's familiar or is aware of keto diet? Rika, have you heard of keto diet? Nagpapapayat? Okay. Can you give me a slight, ano lang, how do you understand keto diet? Ah, yes. Okay. Actually, you have a good point on that one mentioned, Rika. How about the others? Anyone who has an idea about keto diet or might have some friends who have undergone keto diet? Ayan. Miss Katinoy said, high fat diet. Yes, actually, that's correct. One of the materials or one of the things that you usually eat when you're undergoing keto diet is hindi ka mag-ordinary Starbucks coffee. So, mag-Starbucks kayo, guys. Hindi siya pwede kasi ang Starbucks is filled with a lot of sugar. Sugar sa creamer, sugar sa lahat na ng sugar, syrup, and everything ando doon sa Starbucks. Kaya out XXX ang Starbucks sa atin kung gusto niyo magkito. However, there is an alternative and you call that as bullet coffee. Okay, ano naman yung bullet coffee? Okay, anyone who knows about bullet coffee or have heard about bullet coffee? Meron ang alam ko nagtitimpla ng bullet coffee is not Starbucks, but it's I forgot yung isa in not Figaro basta yung isa pang coffee place wherein they place butter on your coffee. Okay? So iinom ka ng kape, walang creamer, pero you place butter in it. Okay? So, yun yung iinumin mo. Yan. Coffee Project. Yes, that's correct. Miss Antonio, Coffee Project po. They usually have that one. Okay? So, yan. Now, so usually what happens here is that you actually cause the body to have low sugar. Okay? And then it will actually cause the increase of the ketones in the blood and the urines. And usually, if it's too high, you already characterize this as ketosis. Now, we have another ketosis, which is physiologic ketosis, wherein the normal response to low sugar, low glucose availability due to diet and fasting provides additional energy source for the brain in the form of ketones. So in physiologic ketosis, ketones in the blood are elevated above the baseline, but the body's acid-base homeostasis, when you say acid-base homeostasis, I mean the pH level is not affected. Okay? Kasi nga, remember, pag ketones, it can actually change the pH level if it's too long that you're undergoing ketosis in the body. Kaya usually, guys, if in case you're gonna be interested in doing keto diet, maganda siya. I had a friend who did keto diet. Puwede kayong ano, as in no carbs, walang kanin, walang kahit na anong sugar, bawal din ang chips, bawal din ang kahit na anong, may starch. Usually magbabasa ka ng labels nun talaga, as in out talaga siya. Puwedeng fatty siya, fatty food but not starch-rich food, okay? So yan po. Pero, you are only allowed to stay under ketosis or keto diet for a certain number of days. And after that one, you have to go back. Kasi nga po, it can have an adverse effect if you're gonna stay on the diet too long. Okay? Okay, so ketogenesis, this is actually the pathway for the metabolism of ketone bodies. So here, your fatty acids and certain ketogenic, okay, we say ketogenic, these are the ones, amino acids that can produce ketones, are broken down and in order to derive energy. Okay, so our body continuously produce ketones in low amounts, but in certain cases, even in case nag-diet kayo, okay, or nag-exam, nag-experiment si Ma'am Santiago for a long time at nagutong, let's say si Norjana, hindi nakakain ng umaga, tapos late na dumating sa klase, tapos gutom na gutom na. Yan. So usually that's when your sugar or your glucose level goes down. Okay? So yeah. So this the ketogenesis would start to work as an alternative energy source already. Okay, so usually this can also happen for healthy individuals when they increase the concentration of fatty acids or carbohydrate, when their carbohydrate reserves are decreased, and then that's when ketogenesis happens at a higher rate. Okay. Yeah. So these are the conditions. If there are low sugar level because of starvation and fasting, yan, exhaustion of carbohydrate reserve. Kumbaga, konti lang yung inagahan ninyo, tapos alternative source like glycogen has already been consumed, yung breakfast, nag-late lunch kayo, yan. And then, if in case you're suffering from type 1 diabetes, okay? So, remember, type 1 diabetes is different from type 2. Type 2 is something that can be developed over time. Type 1 is usually, is this hereditary or not? Let's ask, Sir John, is type 1 diabetes hereditary or not? Or is it developed? Sir John, are you still there? How about Gab? Yes, Ray, Mr. Defa. Anong yes? Hereditary. Okay, so hindi lahat thank you for answering. Not everybody is gonna have type 1. But the most prevalent cases right now is a type 2 diabetes. Okay, alam mo yon, kain pa more, lafang pa more. Now, take note that all the body parts, such as the brain, the muscle, the heart, can utilize energy from ketogenesis. Okay? So yan, take note. All of these parts can get energy from ketogenesis. Insufficient gluconeogenesis results from hypoglycemia, excessive production of ketone bodies resulting to fatal condition called ketoacidosis. So this is actually prolonged ketogenesis na. Okay? And it can be dangerous. here. So here are the different steps for ketogenesis. So usually this is happening in target cells, which is the mitochondria of liver cells. So usually the steps is the first one, um, condensation of 2-acetyl coenzyme A forming acetoacetyl coenzyme A using enzyme thiolase. So coenzyme A is removed from the first acetyl coenzyme A, causing them to be synthesized together to form acetoacetyl coenzyme A. Now the second step, okay, so here you have your acetoacetyl coenzyme A. So this is catalyzed by beta hydroxy. Um, we produce now your beta hydroxy beta glutarate coenzyme A with the uh, with the action of HMG coenzyme A synthase. Okay, so usually what happened here is that water is added to acetic coenzyme A to the beta carbon. Sasama yung beta carbon, so dito siya. Okay, and this produces beta hydroxy beta methylbutyryl coenzyme A, or what we call HMG-CoA, which is a branch 6 carbon compound and intermediate of the synthesis of cholesterol in the cytosol. So yan, so here siya. Then the third one, acetyl coenzyme A, one acetyl coenzyme is cleaved by HMG-CoA lyase, producing acetoacetate. Okay. Then acetoacetate now can be either going to these two products, which is the formation of acetone using non-enzymatic decarboxylation enzyme, or we can form now the D-beta dehydroxybutyrate. Okay. Now take note that this part, or the formation of D-beta hydroxybutyrate, is reversible, while for the formation of acetone from acetoacetate is irreversible. And also take note that of the um, oxidation of your NADH. Okay. Okay. Now, why is ketogenesis important? So, unang-una, it is used to give energy to the brain, the heart, and skeletal muscles under fasting conditions. That's why we can still work, we can still move around kahit na nagugutom na kayo. Okay. Then, the ketogenic diet, which is low carb, fat rich diet, is used. Again, it's a fad right now to lose diet. The idea is to utilize excess fat stored in the body to get energy, but excess ketone bodies can lead to complications and ketoacidosis, yung mismong elevation ng acid sa ating system. And then, in ketoacidosis condition, the kidneys excrete extra ketone bodies with the water resulting in fluid loss. So, nakakapayat siya kasi nauubos yung inyong fat, and at the same time, yung water na nare-retain sa katawan din natin. Okay? Now, diabetic patients are greatly affected by ketoacidosis because insulin hormone is the main regulator of the process. So, medyo delikado to for those who are diabetic. Okay? Yan. And then symptoms, yan, of ketoacidosis, frequent urination, yung fruity smell or acetone ng breath, hindi sa ano ha. Then nausea, pag nasasobrahan na. Shortness of breath, fatigue, and excessive thirst. So that is signs showing that you're already having ketoacidosis. Then level of ketone bodies present in the body can be tested by blood serum or urine sample analysis, which is what we're going to be doing after next Wednesday, not this coming Wednesday, because of the holiday. So wala tayong laboratory for this week. We're just gonna do that next week, okay? So we will have somebody we will be testing urine, okay? Yan. Okay. So any questions so far with ketogenesis? Okay. I guess none. Ira, question. Thank you again. Thank you. Okay. So I have here another video for the ketone metabolism.
In life, it's helpful to have a plan B in case plan A doesn't work out. In terms of energy, the body's plan A is to generate energy from carbohydrates, fats, and proteins, basically in that order. But if these main fuels aren't readily available, then plan B is to use an alternative fuel source, ketone bodies. Ketone bodies are a group of carbon-containing molecules produced by liver mitochondria using a 2-carbon molecule called acetyl-CoA. The liver makes ketone bodies in physiologic states like prolonged fasting or exercise, as well as in pathological states like type 1 diabetes mellitus or alcoholism. Ketone bodies can be released into the circulation and get picked up by the majority of cells. Inside the cells, they're reconverted back into acetyl-CoA, at which point they can enter the mitochondria and produce ATP. The 3 primary ketone bodies are acetoacetate, beta-hydroxybutyrate, and acetone. Alright, so let's say that you decide to go on a 5-day fast. About 12 hours into your fast, your blood glucose levels start to dip. In response, glucagon is secreted from the pancreas and stimulates hepatic glycogenolysis, meaning that the liver starts to break down glycogen into glucose and release that glucose into the blood. About 24 hours into your fast, your liver starts running out of glycogen, so it starts the process of gluconeogenesis, which is when it makes new glucose molecules from substrates like amino acids. Then, around 1 to 3 days into your fast, your body starts to run out of the necessary substrates to make new glucose. So it switches to breaking down fatty acids for energy. Fatty acids are mobilized from fat stores and are broken down to acetyl-CoA through beta oxidation in the mitochondria of most cells, except for brain cells. See, the thing is, fatty acids can't cross the blood-brain barrier, so brain cells can only use glucose for energy. Or when there's no glucose, they use ketone bodies. This makes sense from the liver's standpoint as well, because normally acetyl-CoA combines with oxaloacetate in the citric acid cycle to make citrate. But since oxaloacetate is also a substrate for gluconeogenesis, its levels are pretty depleted at this point in starvation. So oxaloacetate basically leaves all that acetyl-CoA hanging out by itself. Not cool, acetoacetate, not cool. This means that the liver is practically overflowing with acetyl-CoA, and the liver converts it into ketone bodies that various cells in our body, including the brain cells, can use. Ketone body synthesis starts with 2 acetyl-CoA molecules getting joined together by the enzyme acetyl-CoA acyltransferase. The result is a 4-carbon molecule called acetoacetyl-CoA, and then a free CoA molecule. Next, the enzyme HMG-CoA synthase combines acetoacetyl-CoA and acetyl-CoA to form a 6-carbon molecule called 3-hydroxy-3-methylglutarol-CoA, or HMG-CoA. So 3 acetyls and then a free CoA molecule. This step with HMG-CoA synthase is the rate-limiting step of ketone synthesis. In other words, the rate of this reaction determines the overall rate of ketone synthesis. It's like the slowest step in the assembly line for a factory. Next, the enzyme HMG-CoA lyase removes acetyl-CoA from HMG-CoA, leaving behind an acetoacetate molecule. So 2 acetyls and no CoA molecules. Acetoacetate can also be converted into another ketone body. This happens when the enzyme beta-hydroxybutyrate dehydrogenase takes hydrogens from a nicotinamide adenine dinucleotide, or NADH molecule, and gives them to acetoacetate, forming beta-hydroxybutyrate, our second ketone body. However, acetoacetate can also enter the bloodstream. At this point, some acetoacetate molecules remain in that form, and some acetoacetate molecules spontaneously lose a carbon, no enzymes required, to create acetone, our third ketone body. But this acetone is metabolically useless to cells, so it's simply exhaled from the lungs. That's why in pathological states like diabetic ketoacidosis, the breath smells kind of like fruit, and that's the smell of acetone. Now, these ketone bodies are acidic molecules that like to donate their protons, so they can lower the blood pH, causing a metabolic acidosis. Once beta hydroxybutyrate and acetoacetate are in the bloodstream, they can diffuse into the mitochondria of peripheral tissues, like the brain, skeletal muscles, and kidneys. One cell that cannot use ketone bodies is the red blood cell, because it does not have mitochondria. Initially, beta hydroxybutyrate is converted back to acetoacetate by the same enzyme, beta hydroxybutyrate dehydrogenase. This time, though, the enzyme takes hydrogen from beta hydroxybutyrate and gives it to NAD, making NADH. Next, acetoacetate needs a CoA molecule to be converted to acetoacetyl CoA. But unfortunately, peripheral tissues don't have a lot of acetyl CoA, so the enzyme thiophorase grabs it from succinyl CoA, resulting in acetoacetyl CoA and succinate. Ironically, liver cells do not have this thiophorase enzyme, so they can't use ketone bodies for energy. They can only make them for other cells. This is a great example of how the liver is a humble servant of the body. Finally, an enzyme called beta ketothiolase cleaves acetoacetyl CoA, and then adds an extra CoA, making two acetyl CoA molecules, which can then enter the citric acid cycle to make ATP. Alright, as a quick recap, ketone bodies like beta hydroxybutyrate and acetoacetate are an alternative source of energy in states of prolonged starvation. Ketone body synthesis happens in the liver, where excess acetyl CoA from fatty acid breakdown is used as a substrate. Once synthesized, ketone bodies can leave the liver and enter the peripheral cells like the brain, skeletal muscles, and kidney, where they can be reconverted back into two acetyl CoA molecules that can be used to make ATP.
Okay. So, yan po. Wait lang ha. Exit ko lang siya. Yan. Okay. Ayan, guys. Sorry, nahuli pala tong fatty acid biosynthesis ko, yung fatty acid synthesis. This is the fatty acid synthesis. Yan. So, fatty acid biosynthesis is the process of combining 8 carbon uh, acetyl groups from acetyl coenzyme A to form now your 16 carbon saturated fatty acid palmitate. So, the palmitate can be modified to form other fatty acids, and they can include chain elongation up to 18 carbon stearate, desaturation, unsaturation of fatty acids. Okay. So take note, as mentioned earlier, this is happening inside the cytoplasm of liver, adipose, and the central nervous system, mammary gland as well. Okay? So yan. So this is already the part na ginagawa na natin. So, it's learning check again. Okay? So, for learning check, again, have your hands ready sa in-call messages. We will be recording your answers. Okay? So, you have only 30 seconds to answer these short questions here. Para lang malaman natin kung if you're still there, awake, alive, and kicking. Okay? So, yan. So, the first question is, okay, so it is a process of combining two 8 carbon fragments or acetyl groups from acetyl coenzyme A to form a 16 carbon saturated fatty acid palmitate. Again, so this is the process of combining 8 2 carbon fragments of acetyl groups from acetyl CoA to form 16 carbon saturated fatty acid palmitate. Okay, go. Pero lakas. Sige, sulat lang po. Okay, so lahat ba nakasulat na? Baka may delay tayo sa webcast natin. Okay, so let's double check. Okay, so more than 40, we're 45 right now. Yan. Okay, so the answer is And fatty acid biosynthesis or fatty acid synthesis. Okay, so either of the two answers is correct. Okay, next question. So second question out of four. Yan. So it is a catabolic pathway of metabolism, and in this process, fatty acids and certain ketogenic amino acids are broken down to derive energy by alternative means. Okay, so it is a catabolic pathway of metabolisms, and in this process, fatty acids and certain ketogenic amino acids are broken down to derive energy by alternative means. Okay, go. Okay. So, wala na sumasagot. So, the answer is ketogenesis. Okay, third out of fourth question. Okay. So, when an odd number fatty acid undergoes beta oxidation, it forms acetyl coenzyme A, and sorry, it's acetyl coenzyme A, and what? Okay, so it forms now. Yeah. So this is regarding the experiment that was done by Dr. Knoop at the start of the discussion. So it's acetyl acid and benzoate. Okay, so yan. And the last question. Ay, teka lang guys, wait lang ha. Pareho yung question ko. Ay, my bad. Pareho yung question ko. Pareho lang. Sorry, no, my apologies. Pareho lang yung question ko pala. Okay, so that is three questions all in all. So, sino nakakuha ng lahat ng tama? Taas nga ng mga kamay. How many got all three? Okay. Good, good, good. Yes, Irish? Medyo grounded siya. Ayan. So, a lot of you got it correctly. Good, good, good. That's good. Okay, thank you. You can go back and lower your hands, please. Okay, so let me just go to our TBL Hub. Sana hindi ako na-log out. Wait lang po. Okay, so for with lipid metabolism, the only activity that we have for this week is just your quiz. Although we can still open some of your activities na na-miss out. Okay, ayan. Yep. Ayan. Okay? So, yan po. Although yung mga videos here are already incorporated in our TBL Hub or in our PowerPoint that we use today. Okay? Yeah, cholesterol, fatty acid. Okay, so your exam is going to be available later on. Okay? And then you have a week to work on this. Okay? So, any other concerns or issues that you'd like to discuss, guys? before we end the session? Yes. Okay, none. Okay. Sige. Um, Thank you, Nojana. Okay, for confirming that as well. So if you have no more questions, I'll go ahead. You can all now leave the session. We have a discussion with my department head. Okay, I'll see you on Monday, guys. Okay, bye. Thank you. Bye bye. Thank you. Teka lang. Ang galing ko na si Verano. Teka lang si Brent. Teka lang. Leave ko lang to. Oh, ayan. Okay.